Alrighty, thank you everyone for joining us here today for this joint webinar introducing LI-COR's newest plant physiological product, the leaf trace gas integration system, known as the LI-6878. I am Seton Bockel, the plant physiology product manager here at LI-COR, and I will be serving as a moderator and a panelist during today's webinar. And I'm joined by fellow LI-COR principal scientist, Dr. Doug Lynch, who will be presenting a portion of today's material and also be serving as a panelist. Today's webinar is unique in that we are also joined by several members of Asaph Cousin's lab who will be serving as both presenters and panelists throughout the webinar. So this webinar will be split into two parts, the first part being led by Dr. Lynch, and the second half will be led by several members of the Cousins lab who are all pictured here. So with that, please put any questions in the Q&A section and we will get to them throughout the presentation. There will be time for questions at the end, but if you have something pressing, please put it in that Q&A. Thank you. Go ahead, Dr. Lynch.
All right, thank you, Seton. So let's go ahead and dig into what we're talking about today, which is the LI-6878, or the Leaf Trace Gas Integration System, which is a mouthful for describing this new set of hardware. What we're trying to accomplish here is to integrate our LI-6800, the leaf level photosynthesis system, with our what we call our trace gas analyzer platform that has a number of different analyzers for different gases of interest. And the three kind of interest here are pictured here: the LI-7810, which measures methane, the LI-7820, which measures N2O, and the LI-7825, which measures carbon isotopes. So this kit basically pairs one of these analyzers with some additional hardware that I'll go into detail here on the next slide to provide a fully integrated system for either fluxes at the leaf level of methane or N2O, or for quantifying carbon isotope discrimination. So this kit kind of fully put together, our intention here is to provide a complete solution. And to us, what that includes is all of the plumbing required. So there is a new manifold that bolts onto the front of the LI-6800 sensor head, allowing access to sample the exhaust airstream. This kit includes all of the tubing required to get that airstream from the 6800 to the TGA. And it does so via this box here, which I will go into detail in on the next slide. What that box is doing is allowing, or its main component is dealing with water vapor, which is a serious consideration here. And it also allows a user to incorporate a couple of calibration tanks, which can be important for these types of measurements. We're also providing a full software solution such that the data stream from the gas analyzer of choice is integrated automatically into the user interface on the 6800 and into those data files, which I'll show in detail on the next slides. So just to be kind of transparent here, right? If you've attended or have an LI-6800, you're familiar with our measurements of leaf level gas exchange, which requires a concentration measurement in two places: the reference before the airstream interacts with the leaf, and the sample after the airstream interacts with the leaf. In this case, we have only one analyzer for this additional measurement. And so we have to measure the reference and the sample at different times. And we do that because of the way our sampling scheme is constructed. When the LI-6800 goes into match mode, that is when the third analyzer will receive the reference airstream, and then leaving match mode, now it will be sampling the sample airstream. So we're able to capture both the reference and the sample at slightly different points in time. And that's an important consideration for these measurements. Oops. So a little more detail on what this additional hardware is doing that can attach to the side of your console. So what it is what it's doing is you can see from this scrub tube on the side full of our desiccant, or you can supply your own dry air stream. It is running that air through a drying system and removing much of the water vapor that's in the airstream. And that's because, again, we're sampling from both the reference air and the sample air. And due to the addition of water vapor via transpiration, those two airstreams have very different water vapor concentrations. And that causes issues in the TGA measurement. So we want to remove that water differential, and that's how we're doing so, by running it through that drying portion. The other capabilities that this dryer box provides, as I mentioned earlier, is the ability for the user to connect a few calibration tanks, and those calibrations can be automated as part of the workflow if the user so desires. Another part of this solution is the software integration. So here's an example using the LI-6878 for carbon isotope discrimination. And sort of in real time on the user interface, we have these discrimination parameters available for plotting. This discrimination _I, or for infinite, is the theoretical discrimination one would expect if CI equals CC, or there is no resistance to movement of CO2 through the mesophyll. This sort of moving line here is capturing the actual measured discrimination by this system, which again, we're calculating on board in real time. And so the user can in real time see how much discrimination is occurring and if their measurements are valid. And over here, I've highlighted that we're also able to calculate mesophyll conductance on board as well in real time. So you can see those calculations as the measurements are happening. So just to take a step back, we'll want to mention that there are two separate, unique applications here that we see. The first is the one I kind of just went over a little bit, the LI-7825 providing carbon isotope discrimination. And that's what the Cousins lab will be providing a lot of measurement examples on. They have spent a lot of work validating that measurement, and we'll be hearing from them here in a few minutes. The other application is for trace gas fluxes of methane or N2O. And those are both available, same hardware set. The only difference is which of those third analyzers are part of the system. And the software will automatically adapt here, as I'll show you in a minute. So this slide is, what would people use these for? For methane, methane is an important greenhouse gas. So understanding those fluxes mediated by plants is an important research topic. Same with N2O. There's a lot of agricultural applications that may want to quantify N2O fluxes through leaves. And these plots here on the bottom are showing at a few different flow rates, sort of our minimum detectable flux as a function of flow rate. And on the x-axis is the leaf area. So one thing this highlights is that the larger chambers are probably going to be most appropriate for a lot of these measurements. And that will come up again with the isotope discrimination. This is not a standard LI-6800 measurement. The sort of demands of this measurement are a little bit more complicated. And you really need to think carefully about if your measurement system is appropriate for these, based on the amount of leaf area you can fit in that chamber and the fluxes you expect for either methane or N2O. Moving on to the sort of carbon isotope discrimination, the LI-7825 is able to measure the 4 most abundant isotopologues of CO2 noted here. So we're measuring the sort of major isotopolog, 12 CO2, and then 3 of the minor isotopologues, the CO2 that has a heavier 13 carbon, and then the 2 heavier oxygens, 18O and 17O, are also being quantified. And this plot here is showing at a few different CO2 concentrations, our noise or standard deviation in that reported delta 13C measurement, again, as a function of averaging time. And that's another key point here is the averaging times that you're used to in a LI-6800 measurement are probably not appropriate for a lot of the measurements with these trace gas analyzers. You're going to need a significantly larger averaging time to make these measurements, or to get the noise to where you can make these measurements reliably. Sometimes on the order of up to a minute or even longer, up to 2 minutes perhaps of averaging. So these are there's a little more complexity to these measurements that have to be considered. And this figure on the right is just showing, I really like this figure from a paper by Clark et al. In 2020 that sort of shows what's happening to a CO2 molecule as it works its way from the intercellular airspace into the site of where the chloroplasts are. And it shows all the sort of pathways that can lead to resistance to that movement. And that's essentially what is being attempted to quantify with these discrimination measurements. So these figures on the right are showing the sort of summary of the computations that we are providing on board the LI-6800, as well as kind of an idea of at a given flux, the amount of leaf area that is necessary to get the error rate into an acceptable level. So this top plot here is showing essentially, it's a parameter called psi. It's a measurement of the drawdown of CO2 in your cuvette, which is a function of both the leaf area, this x-axis, and the assimilation rate. So at higher assimilation rates, you can get essentially lower error is basically what this is saying. But if you had a leaf that had very low assimilation rates, the errors are going to be much larger, and this measurement may not be appropriate. So it's something to consider. We're going to be drying the airstream before it enters our TGA. We're going to be using additional consumables, either dry-rite or silica gel, whatever desiccant you use. You go through quite a bit of it. So that's something to consider. As I'll show in the software here, the LI-7825 has some CO2 dependency to it that we are attempting to quantify and remove. And that's a big part of the software package that we're providing. I already harped on this, but averaging times are going to get quite longer, and that's something to keep in mind. And those calibration tanks are useful to make sure your isotope log analyzer is responding as expected. So let's take a quick look at the software. So our software that we're about to launch here will have this new option to support TGAs. And if you choose to check that box, you get a brand new tab into your workflow. And then you can connect to your given analyzer, which you would select the model and make in this tab. And then once you've connected, it gives you kind of some status information on that analyzer, right? It gives you its battery life, it gives you any diagnostic info, and tells you the information about the concentrations it's reporting. All of these trace gas analyzers stream data at 1 Hertz, 1 data point per second. And that's being integrated automatically here into the LI-6800. Moving down in that TGA tab, we have this source and calibration, gives you information on the status of that, as I mentioned, that dryer box, which is depicted here, and the flow going towards the TGA. It allows you to turn on and off calibration tanks here. That can also be automated as part of your workflow. There's also some updates to our environmental control screens. You can see here in the flow screen, we have real-time information on where that flow is going, be it through that dryer box to your TGA. Again, we're sampling from the exhaust of that sample gas analyzer. So it's telling you if you have sufficient flow there, and that flow is actually moving towards a TGA. So it gives you kind of some useful diagnostics. That CO2 dependence can be removed by basically running a series of CO2 concentrations and capturing how much that delta 13C is changing as a function of CO2 on a known constant background. And we're able to capture that and correct for it in real time. And this is a procedure that has to be run frequently, probably a couple times a day, perhaps, depending on the measurement campaign. And again, this is showing real-time calculations of important parameters, including the discrimination against 13C and mesophyll conductance, which is an important parameter a lot of people are going to be interested in. One last slide here for me is this is not a new feature. We've had these background programs as part of the LI-6800 software for a few years now, but they're going to become, I think, more critical with this application. And you can write your own custom programs to perform a series of automated steps. And the data about to be shown to you by the Cousins lab used two of these background programs to do light response curves and CO2 response curves. So they were able to select the light values or CO2 values they wanted to measure. You can see there's potential for needing to wait longer at these step changes because now you have this additional averaging time to incorporate. And they wanted to get multiple data points at each of their set points and to kind of get an idea of the noise and to quantify maybe over time as well. And this program allowed them to do so. So they were able to capture basically 10 data points at each of their set points via this type of program. And it's fully automated. And these are extremely flexible and I think will be very useful for these new applications. So I think that is the end of my sort of intro section. And now I'm going to hand it over to, I think we're going to start with Haley, who is going to start talking about some real actual data they've collected with this system. So Haley, go ahead and take over.
Yes, hi everyone. I'm Haley Schroeder. I'm a graduate student in the Cousins lab here at Washington State University. And I'll just provide a brief introduction to our plant material, our measurement conditions, and the tests that we've conducted so far on the LI-6878 system. So I do want to note that the measurements that I'll show today, or that we'll all show today, are on growth chamber grown plants. And I'll start by showing some data on the C4 model graph Soteria viridis. I have our growth chamber conditions and the plant age during measurements detailed here, but if you're interested, you can read through that. I won't go through the details. But after my introduction, Rita, a staff scientist in our lab, will show you her measurements on C3 rice plants. And finally, Diego, a graduate student in our lab, will show you his work on the C4 maize and some Soteria viridis measurements as well. So we took our measurements, like I mentioned, inside a growth chamber in the lab with a temperature of 30 degrees Celsius using the 3x3 chamber. For our standard conditions, I will note that our Soteria viridis and maize data was taken at 21% oxygen, while the rice data was taken at 2% oxygen to minimize photorespiratory conditions. We were interested in measuring leaf photosynthetic responses to light and CO2 with this new equipment, and I want to note that the measurements were taken in sequence, with the light response curves followed directly by the CO2 response curves. So starting with the light response curves, we set a CA of 400 micromoles per mole, and our light scheme started with 200 and ended with 1500 micromoles of photons per meter squared per second. We measured each light condition for 10 minutes, as Doug mentioned, with logs taken every minute, and the LI-6878 matching occurring after logs 2 and 7. Our light response curves served as the acclimation for our CO2 response curves, which were taken directly afterwards. And these were our CA schemes for rice and for Soteria viridis and maize, with the same 10 minutes at each condition with 1 log per minute. And like I mentioned, we use the BP Builder program written by Doug to run these measurements. So as a relatively new LI-COR user, and I'm relatively new to gas exchange measurements in general, I wanted to first practice using the LI-6878 to take light and CO2 response curves with Soteria viridis. So here I'm showing the response to light with 9 biological replicates plotted. And next I'm showing you the response to CO2 with those same replicates. And my takeaways from this test were that the variability between replicates was really minimal, and the precision of the LI-6878 shows that the equipment's not disrupting gas exchange. Also, given that I don't have, or that I have some but not a lot of experience using LI-6800s, I would say that this equipment was very easy to learn and use. So to test the LI-6878 compared to theoretical values and previous data from other systems, we can plot the discrimination that we collect as big delta 13C versus CI over CA. And I just want to introduce this figure because it shows how we'll use these variables to represent our data in the coming slides. So the CI over CA tells us the ratio of intercellular CO2 to ambient CO2 for the leaf. And this is the simplified model of discrimination compared to CI over CA for C3 plants. And we can see that for C3 plants, as CO2 availability increases, so does discrimination. For C4 plants, this is the simplified model. And in this case, we can see that discrimination decreases with CO2 availability. And I also want to highlight that in C4 plants, discrimination is typically quite low, which makes this a more technically challenging measurement to take. So as I mentioned, our goal was to test this LI-6878 system in terms of how carbon-13 discrimination responds to both light and CO2 availability. And with that, I'm going to transition to 2 other Cousins Lab members. First, Rita will show you her tests on a C3 plant, followed by Diego with a C4 plant. And give us just 1 minute to transition over.
Hello all, I am Rita Giuliani and I'm showing you our results using the new stable isotope trace analyzer on rice. The species is Oryza sativa and is a C3 species, so C3 photosynthetic machinery. Wait a moment, please. You can assist me. Thank you. The first data are for the leaf photosynthetic analysis in response to the incident light. This is a response of the leaf net photosynthetic rate to increasing light levels, ppfd from 200 until 1500. This is the corresponding leaf 13C discrimination. And we observe a decreasing pattern with the increasing light levels. The standard error of the measurements are reasonably low, and for almost all the light levels, for the first low ones, we see a higher standard error, higher variability that is associated to a low isotope signal compared to the intrinsic noise of the equipment. The intercellular to atmospheric CO2 concentration plotted versus the light levels show a decreasing pattern, and the decreasing pattern is explaining the decreasing trend we observe in the 13C discrimination we just saw. When we plot the values for the single biological replicates of the 13C discrimination, we see in the plot upper right on the right, plotted versus CI over CA, we see they display an increasing pattern that is reflecting the increasing trend by the theoretical model of the 13C discrimination versus CI over CA. We did a comparison between the 13C discrimination by the new tracer compared to a reference technique that is the TDL-AS, is a spectroscope used in the lab. It was the first technique available to investigate stable isotopes in leaves. I put some literature. The first one is 2003, actually, is describing the system, the original system. All the other publications come from the research in Dr. Cousin's lab during the years. Okay, we see here a comparison between the 13C discrimination obtained by the new tracer in with red dots compared to the TDL data in gray. We have two experiments providing the data for the new tracer. You have the red full dots and the empty dots. And in general, all the data define this, however, follow this increasing trend of 13C discrimination versus the CI over CA that is expected and as indicated also by the theoretical line. We see that the variability among the single replicates, biological replicates for the new tracer data, the variability is not higher compared to what we have in the TDL measurements. And this is a positive response. We analyzed the leaf photosynthesis in response to CO2. We have here the response of the leaf net photosynthetic rate to levels of atmospheric CO2 concentration from 300 to 1500 in molar fraction. The decreasing values you see after 800 CA is not related to the equipment, is a biochemical limitation. We see across all the CO2 levels, however, a reasonable variability in the data. The equipment seems to work, to operate correctly. And we have the discrimination, leaf 13C discrimination for this data, increasing trend with the atmospheric CO2 level. We have the intercellular to atmospheric CO2 concentration plotted versus the atmospheric CO2 concentration, and that there is an increasing trend, and the increasing trend explains the increasing trend we have in the plot on the right, above, top right, for the 13C discrimination versus CI over CA. We plotted the 13C discrimination versus the CI over CA. We see increasing trend of the values that reach almost the theoretical increasing line. We made a comparison again between the values by the new tracer and the reference technique. These are data per single biological replicate. The red dots are for the new tracer and are coming from the experiment before. We see that again, the data of both equipments tend to build this increasing trend that is expected based on the increasing theoretical line. The new tracer is providing values of the mesophyll conductance, mesophyll CO2 conductance. This plot, it was just to test if the inputs for the model that we enter into our spreadsheet and the equipment were providing the same data. It's just an empirical validation of the calculations by the new tracer, because the model we use in the lab is essentially the same one based on events and phone camera. Maybe in the new tracer, there are some adjustments implemented based on UBR and the colleagues later, but it's the same model. So we say, yes, the calculations are fitting what we expect. And here we have the absolute values we determined with the new tracer for the mesophyll CO2 conductance over the several light levels. Again, we see low variability in the data. We have 6 biological replicates, so high precision. And compared to the reference technique, okay, the absolute values are a little bit different, but the variability is in the same range. So this is positive. And then the mesophyll CO2 conductance versus the atmospheric CO2 level, decreasing expected trend, but again with low variability in all conditions. The new tracer is able to detect also the oxygen 18 signal in CO2. Here we have the response of the discrimination for 18O in CO2 versus the CI over CA. The dashed line corresponds to the theoretical line, and is in analogy to the theoretical line we observed before for the 13C discrimination versus CI over CA. Here is not a linear pattern, is a curvilinear, and all the squares are data collected per single biological replicate using the new tracer, and they cover a range of CI over CA that is from 0.65 to 0.90, generated based on different levels of CO2 set in the equipment, from 300 to 1500, essentially the same CO2 levels we have seen before. And we see that the data tend to follow this increasing pattern. And if we add, these are mean values of comparable experiments. When I say comparable experiments, means the same plant material under the same environmental conditions. We have 3 TDL experiments, and these are the mean values. And we can consider altogether all of these data following this expecting increasing pattern. And so positive. In a positive way. In summary, the use of the new LI-COR tracer in C3 species showed precision under a range of light and CO2 concentration levels. And we see accuracy in the 13C discrimination and in the discrimination of oxygen 18 in CO2 compared to the reference technique. And we see precise leaf mesophyll CO2 conductance. And what is very convenient is that mesophyll conductance is provided in real time. And this is my summary, our summary for the C3 test of the equipment. And next speaker is our colleague Diego for the C4 part. Thank you.
All right. Hi, everyone. Again, my name is Diego Zamudio, and I'm a graduate student at Washington State University. My part will focus on sharing some data where we collect carbon-13 discrimination into important C4 species, Cetaria and maize. Next, please. As we observed before, in the case of citrus species, we know that the carbon-13 discrimination is positively correlated with CI over CA. While in the case of C4 species, we observe that the relationship between CI over CA and carbon-13 discrimination is negatively correlated. This relationship makes C4 species not only to have lower carbon-13 discrimination values than citrus species, it also makes carbon-13 discrimination challenging to measure. Even though carbon-13 discrimination in C4 species is not easy to measure, it is widely used to study C4 photosynthesis. To test this new system, we use maize genotypes that we know that they have next please, that they have a similar assimilation rate, or A, under different line intensities. Next, they have a different GS, or stomatal conductance, under different line intensities. Next, and as a result, both genotype they differ in their CI over CA values. So we already know this. And now our part was to test if this system is able to detect those differences, but in terms of their carbon-13 discrimination. And indeed, in the four graph, the one that is on the bottom right, we observe that the system is able to pick up those differences. Next, please. So now, if we plot the carbon-13 discrimination versus CI over CA, next, this graph, it confirms what we already seen before, that the differences in carbon-13 discrimination is driven by differences in the CI over CA values. And the values follow the trend of this theoretical solid black line. Additionally, next. Additionally, we added previous data that was collected in 2022, but in this case, they use a different tool known as the TDL, and we can clearly see that both data sets, they overlap, which give us a good idea of how precise this new system is. Next. So, and in addition, we wanted to test also if this system is able, to provide reliable carbon-13 discrimination values under different CO2 concentrations. And we test that, but in this case, we use the second C4 species, Cetaria. And here in the first graph, we see that the system provides reliable assimilation rate values. It doesn't interfere, this new system doesn't interfere with the gas exchange data. Next. And also, again, it also provide us with, or this system is able to detect differences in the carbon-13 discrimination at different CO2 concentrations. Next. Again, we wanted to plot the carbon-13 discrimination for this species versus CI over CA. And again, we also, it shows us that the differences in carbon-13 discrimination is again driven by differences in the CI over CA value. And again, they follow the trend of this theoretical line. Additionally, we plotted previous Cetaria data that was collected in 2017, and just to see how similar both data sets are. And although both data sets don't overlap, both follow this theoretical solid black line. Next, also, I want to point out that this high carbon-13 discrimination value shows how challenging it is to measure this variable in C4 species, because especially at low CA over CA values, any extra noise can disproportionately affect the calculations on the carbon-13 discrimination. Next. So to sum up, this new device, the LI-6878, measures reasonable carbon-13 discrimination for both C4 species, maize and setaria. It also detects differences in the carbon-13 discrimination in maize genotypes with different CI/CA values. And also, the carbon-13 discrimination is still challenging to measure, especially at low CI/CA values. Next, please. As we previously observed in the RISE data that was shown by Rita, where we observed that the N2O discrimination of CO2 increases as the CI/CA value increases, we wanted to see if our maize data, follow also this theoretical trend. And indeed, we can clearly see that both maize genotypes, they follow this trend. And again, this gives us the idea, or this is telling us that this system is able also, to provide reliable values in terms of the N2O discrimination of CO2. Our overall summary is that this new device is a user-friendly tool that provides carbon-13 and N2O discrimination measurements with gas change data while integrating both outputs from the LI-6800 and the carbon isotope in a single file. It also provides reliable values for the carbon-13 and N2O for both species, C3 and C4. And overall, this new device enables both experienced researcher and especially early career scientists to obtain accurate and rapid carbon-13 and N2O discrimination data. With that, thank you. And these are our references in case you wonder, or in case you want to see the data that we have and we plotted in our graphs.
Great, thanks, Haley and Rita and Diego. That's really a lot of really wonderful data to see that we're happy to see collected with this new system. So we've got plenty of time for questions. I think there's been a few in the chat already that I answered that I will start by answering verbally, and I think there's a couple I'm going to pass off to maybe one of you would like to take, but I'll start. The one questions or a few questions I wanted to address to the whole group. One, there was a question about won't the desiccant in that dryer system interact with CO2 and cause a problem with the measurement of the airstream going to the TGA? And I probably didn't explain the dryer in detail enough, but the airstream does not pass through that desiccant. Instead, it passes through tubing of a material called Nafion, which if you're not familiar with Nafion, it is a material that allows the passage of water vapor, but not other trace gases. And so the sample airstream passes through that Nafion, and that desiccant dries the air in the sort of in that small section of air just outside the Nafion tubing. So there's dry air outside that Nafion, and water vapor diffuses through the Nafion, and that's how water vapor is removed. So the airstream never directly comes in contact with that desiccant. There was one other question I wanted to address. Flow rate. There was a good question about the minimum flow rates, and that is an important consideration here. The minimum chamber flow rate is dictated by the flow demanded by the trace gas analyzer, which has a constant flow rate. And that flow rate is nominally 250 SCCM, cubic centimeters per minute. If you're used to 6,800 flow units in micromoles per second, that's roughly 200 to 250 micromoles per second. So that is the minimum chamber flow rate that's acceptable for this use case. If you go below that, the problem is now your TGA will be somewhat getting ambient air, and you won't be measuring chamber air. You won't be measuring what you think you're measuring. So that's an important consideration. One question that I will see if one of you from the Cousins lab wants to answer. There were several questions about water use efficiency. And I know, Diego, you touched on that a little bit, but I think there was some questions about how these measurements relate to maybe water use efficiency from isotope measurements, isotope discrimination on dried leaf biomass, and how all of that relates to these water use efficiency measurements. Do one of you want to tackle the question of water use efficiency?
Do you want to do that? I think you're muted at the moment.
Yeah, we can't hear you, Diego, it looks like you're still muted.
I can go ahead and answer that.
That'd be great.
In the meantime. So, I mean, the instantaneous gas exchange is going to give you, oh, there you are, Diego. Go ahead.
Oh, sorry. Yeah, I don't know what happened.
That's fine.
I mean, well, actually, you know that in the case of water use efficiency, that is a relationship between the ANET and the stomatal conductance. Like actually, the data that I show of maize, those two genotypes, they are different. But one of the distinctions that they have is that they differ in their water use efficiency. So yes, I mean, like basically, you know, the relationship between the water use efficiency and the carbon-13 discrimination, they are inversely correlated. So that's, I mean, that's something that I just wanted to add.
All right. There's some other questions, sort of a number of questions related to calibration tanks and what gases are needed and what is used for a reference. And I honestly don't know if I recall what you guys did to calibrate your gas analyzer. If you did calibrate your TGA, if you did zero, I think you zeroed it on zero CO2 free air. But I don't know if you performed a span in your measurements. Did you span, Rita? It looks like you're nodding your head. Did you guys have a tank of known delta 13C that you ran through the analyzer and periodically calibrated it? No? Okay. So this is a difficult question to answer. Calibration demands here. I will, in my experience, in my hands, this analyzer is fairly stable over time. It does drift a few per mil up to 1 per mil per day, typically much less than that. But it can drift over time. But keep in mind that this measurement is a difference between the sample and the reference. So a lot of the error from an absolute calibration offset disappears in the calculations. It's not zero, but it's pretty small. So there will be a need for periodic calibration of the isotope analyzer. But how often that is really going to depend a lot on the types of experiments being performed. And I would encourage anybody thinking about it to consider how the errors, the absolute offsets, are going to impact their measurements. Because it will matter in some cases and matter less in other cases. Let's put it that way. Often, internally, you know, we know that the background atmosphere is something like -9 per mil, -8 to -9 per mil. And I often will take my analyzer and just have it measure ambient for a while and see if it's close to that value. And I know it's fairly well calibrated.
Yes, so this is the only, I didn't say because it's the only calibration we did to check with the atmospheric carbon 13C composition. I didn't call calibration because, you know, it's just more or less 8 or 9. So I didn't
is it close?
say,
is it
but
believable?
It's what we do.
Yeah, and that's kind of what I've taken to do as well.
We did at the beginning because we didn't know, so we said, let's check with the environment. The other thing only we can say is that we checked the variability you have mentioned during one day, the reference carbon 13 composition has a variability that is even below 1 per mil. over a whole day. Over a few hours, you have actually is 1/5 of that. So it's extremely precise.
Yep, I would agree with that. Sort of corollary to this, there is, and I mentioned this in my portion, but there is a concentration dependency, and that absolutely is real and needs to be accounted for. And so that routine we have built into the software does definitely need to be run. That is an important part of the operation using the isotope log analyzer. Let's see. Let me see if there's other questions that we have not addressed. There's a question about reactive species, mentioning that the 7825 also does measure ammonia. No, the performance of that ammonia measurement is not accurate or precise enough to make any useful measurements in a steady state system, as well as due to ammonia's incredibly sticky, like water vapor. Flush times would be long. It's not a measurement we've attempted to do in any way. Yeah, we discussed calibration a bit already. Let's see, there's some cost and availability questions, and I'm guessing, Seaton, you may want to chime in here on those questions. We'll note and probably reach out to you at a later time. Do you want to address those, Seaton?
Yeah, please reach out to your sales territory manager for those costs and for the timing. It is a relatively lower cost integration between these two like powerful instruments we have in the trace gas analyzers and the LI-6800. But for those costs, and it says please reach out to your request a quote. It is quotable, and it should be available within the coming months. So please stay tuned. You will not get there will not be a lack of LI-COR emails heading your way in the near future regarding this product.
Yep, there's another question about using our CO2 cartridges to check calibration. And yeah, it's similar to checking it against ambient air. Those cartridges within a given lot are pretty consistent within, in my measurements, a few per mil, but there's no guarantee. So I would not trust it as an irrevocable, as a guaranteed calibration source. I would never trust one of those CO2 cartridges for an absolute perfect calibration, because there is some variation cartridge to cartridge. But yeah, they're all fairly consistent near atmospheric values in my experience, but that's never a guarantee because it depends on where they source the CO2 for those. Let's see, there's a question about, yeah, we've been talking about the isotopic measurements a lot here, which is what the Cousins lab does and did for us. And testing on methane and N2O is much more limited from a biological perspective. In our sort of perspective, the demands of this isotopic measurement actually kind of far exceed the demands for those N2O and methane measurements. So we're confident it would work. We don't have, at this point anyway, a ton of biological data to share, but we have kind of tested it in our experience, and it seems to work quite well. Let's see, there's a question about the CO2 cartridge. I'm not sure I understand that question. I'll have to follow up with that one later. What else is unanswered here? Is it possible to use in CO2 flux monitoring simultaneously with gas exchange? Yeah, if I understand that question correctly, I mean, that's essentially what we're measuring here. The flux or assimilation of CO2 of the leaf in the chamber is what's being quantified, and we're just seeing relative fluxes of the different isotopologues. But I'm not sure I understand that question completely. So I can follow up with that person later as well. So some of these questions about, yeah, others that are making these measurements, yeah, we'll follow up with you later on those as well, for sure. Yeah, actually a question about recording the webinar. Yes, this is, Seaton, you want to answer that one? Looks like you were about to.
Yeah, let me get in here. I gotta answer some. Yeah, so there will be the recorded version of this will be made available. We'll be sending that out, and you can always hop back onto the product page on our website and check it out there as well. It will be available. So.
Here's a question. I'll see if anybody from the Cousins lab wants to address. The specific question is about drought stress experiments, and does this improve our ability to separate stomatal from mesophyll limitations? And what's the best workflow to estimate GM under low stomatal conductance conditions? Anybody else want to tackle that? That's a tough question.
Yeah, it depends, Emerita. I think it's yeah, feasible, but obviously when you go under not ideal conditions to have you know, high photosynthetic response, so you need to be patient and to try to find your measurement conditions, so to operate what flow to use, what CO2 to use, what light level, you know. But then I don't see why you couldn't. Just like I say, me personally am used to work with plant material that is not just wild type. And so always I have to spend time to define my measuring condition to be able to detect in the best way the signal I want to get. And so I think it's the same for people working under water deficit conditions, so in drought experiments. And it depends on the species. I don't know what kind of species the plants they use, how old they are. But definitely, I think the equipment can be used for this. Yeah. Not right away, like I say, because yes, it's quite magic, this equipment, but however, it requires time to identify your conditions of measurements. But I think, yes, it's feasible.
Yeah, I guess I'll just add to that to Rita's point that it's not always going to be sufficient, and you really want to think carefully about protocols as well as the chamber. Yeah. You know, leaf area is super critical here. If you're only clamping on a very small portion of leaf, you're not going to generate enough signal.
We have experience where it takes almost more time to define the conditions of measurement compared to the time required to conduct the measurements on a certain number of biological replicates. Or the time can be the same, but then you go smooth measuring. Is at the beginning that you have to pay attention and to set your flow rate related to the light conditions, related to the CO2.
Mm-hmm.
Yeah, okay.
Yep, thank you, Rita. Yeah, there's a question about variations in ambient temperature. And that's something we're still exploring. A lot of the measurements we have made have been in the lab. I have done some measurements in a greenhouse, a hothouse, where ambient temperature is changing throughout the day. The TGA does show a little bit of dependency to ambient temperature that I've seen, and it does require kind of monitoring that, and that isocal routine may need to be run a little more regularly under those changing environmental conditions. That is one caveat that I would say is there may be a little more attention to some of the sort of calibration needs under ambient temperature changes. Well, we have hit our hour, so I think I just want to say thank you to the Cousins Lab and all of the contributions you guys made to both today's webinar and all of these measurements that really helped us validate this. Some lingering questions here we will follow up with people on. I don't know, Seaton, any final words?
No, just thank you to, again, the Cousins Lab and also everyone for attending today. These were some great questions, and as Doug has already mentioned, we'll follow up with these. And yes, you can rewatch this this webinar in the coming days, and always reach out to us with additional questions. Absolutely. So thank you all for attending.